In this lesson, we'll conclude our discussion of neurophysiology by taking a look at the synapse. So we know from previous lessons how the action potential is generated, how it travels down the axon, but now when it gets to the end of the axon, how does it get to another cell? Well, that takes place at the synapse. And then we'll finish this up by taking a look at the cell that's receiving the information. How does it process the information that it's getting? So we call this information processing. So think of the synapse as a specialized site where a neuron communicates with another cell, right? That neuron can communicate with another neuron. It can communicate with a muscle. It can communicate with a gland. The neuron that's sending the message is known as the presynaptic neuron. The one that's receiving the message, right, in the synapse is the postsynaptic neuron. Now, there's two types of synapses. We're going to be focusing on the second one, which is the chemical synapse. So I just want to let you know there are synapses in the body that are known as electrical synapses, where the cells are actually in direct contact with one another. Um, here, just to finish this up, the presynaptic and the postsynaptic membranes are connected together by gap junctions. If you remember from a previous lesson, gap junctions allow for communication between two cells. I remember there was the protein called the connexons, right, and that allowed for they had kind of pores in them, and the materials, such as ions, could pass between the cells. Right? So with electrical synapses, the cells are touching and they're communicating ions between the two cells. And now, as a result, any local current, right, local current is going to affect both cells. Right, the action potentials are actually propagated quickly across these synapses. We find these in some areas of the brain, the eye, and then even in a cardiac muscle. But again, the one we're going to focus on are the chemical synapses. This is the most common type of synapse between neurons, right? It's the only type of synapse between a neuron and another cell. There's no electrical synapses there. They're only going to be chemicals. Now, these cells that are at the synapse have a separation, right? They're not connected, they're not touching. That separation you can call the synaptic cleft or the synaptic gap. And as I said before, this is just a repeat. The presynaptic cell, that's the one before the synapse, that sends the message. The postsynaptic cell, that's the one after the synapse that receives the message. So let's say here, this would be the axon terminal of an axon. Right here's the telodendria. And then in here is where the neurotransmitters are. Notice this axon terminal doesn't touch the postsynaptic cell. There's a space there, right? That gap would be the synaptic cleft. So different types of chemical synapses. We have the neuromuscular junction, right? This was also studied in a previous lesson. This is the synapse between the neuron and the skeletal muscle. But we could also have a chemical synapse between the neuron and the gland, right? So this would be called the neuroglandular junction. And this is how a nerve would, stimulate a gland to release its product. So what are these chemicals, neurotransmitters? There's roughly 90 something, 98, maybe a little bit more neurotransmitters out there. These are chemical messengers contained within synaptic vesicles in the axon terminal. So if we go back a moment in here, these little green structures are actually think of them like a water balloon, but instead of having water in them, they have neurotransmitter in it. We call those synaptic vesicles. Where are we, right? So these chemical messengers are contained within synaptic vesicles within the axon terminal, and that's going to be in the presynaptic cell. During the process of the synapse working, these chemicals are released into the synaptic cleft. They're going to then diffuse across to the postsynaptic membrane where there's receptors, and then that will start the action potential on the postsynaptic cell. We do have enzymes that are going to break these neurotransmitters down. If you remember from, again, previous lessons, there was something called acetylcholinesterase, right, which broke down acetylcholine. And we can recycle the acetylcholine, let's say, in the axon terminal. So it's actually part of the acetylcholine goes back up into the axon terminal and it's going to be recycled. So what are some of the functions of chemical synapses? The axon terminal releases the neurotransmitters that, again, are going to bind to the postsynaptic plasma membrane. There are receptors in that postsynaptic plasma membrane, and then that's going to produce localized changes in permeability, and again, the graded potentials. Again, that's that the local current. The action potential may or may not be generated in the postsynaptic cell depending on the amount of neurotransmitter release and also the sensitivity of the postsynaptic cell. I'm going to talk about this a little bit more at the end of this lesson when we do the information processing. Just kind of accept this for now, that an action potential may or may not be generated in that postsynaptic cell. And we'll talk about that in the information processing. So we have a couple different types of synapses based on the neurotransmitter. A cholinergic synapse is one that releases acetylcholine. And just by the way, if I haven't said this before, the abbreviation for acetylcholine is a capital A, capital C, and then a H, right? Acetylcholine. So it releases acetylcholine at all neuromuscular junctions, right? So every synapse between your nerves and your muscles, that is always going to be acetylcholine. Acetylcholine is released in many synapses within the brain and spinal cord, and then also at all neuron to neuron synapses in the peripheral nervous system. All neuromuscular and neuroglandular junctions in the parasympathetic division of the autonomic nervous system are also going to have acetylcholine. So you can see acetylcholine is at a lot of different synapses. Let's take a look at the sequence of events. So I'm going to skip the words and let's go to a picture. So would everybody, I guess, agree, right? Here's the axon terminal, the end of the axon of the presynaptic neuron. And then over here, this is the cell membrane of the postsynaptic neuron, and it's going to have the acetylcholine receptor. Here's the gap. So the first thing that happens is the electrical impulse travels down the axon terminal. Now, that's going to stimulate calcium to diffuse into the axon terminal, which is important. So we can see calcium now comes into the axon terminal. That prompts these synaptic vesicles, which are filled with acetylcholine, to migrate down and fuse with the cell membrane of the axon terminal and release the acetylcholine into the synaptic cleft by exocytosis, right? By that process known as exocytosis, right? So it's important that that calcium comes in. Once the acetylcholine is released, it's going to diffuse across the synaptic cleft and it's going to bind to the receptor. So here we see the acetylcholine coming to the acetylcholine receptor, and that now opens a gated channel and sodium starts to move into the cell, and now we get a local current, right? As we talked about in previous lessons, right? The local current will now travel along whether this is the dendrite of a neuron or the cell body of the neuron, right? It'll travel through the neuron as a local current until we get to the initial segment of this postsynaptic cell, and then we'll get the action potential. Okay, so here we see again the acetylcholine binding to the receptor. That opens the channels, sodium gated channels, and sodium now comes in, producing a graded potential. Once the neuron stops firing, and we don't want this postsynaptic neuron to be stimulated anymore, we need to get rid of whatever acetylcholine is in the synaptic cleft. So we have an enzyme called acetylcholine esterase. A lot of times they don't even say the acetyl, they'll just say cholinesterase, right? So you'll hear that term also. Same thing, right? No different. That's going to break the acetylcholine down, right? We actually take the choline part that gets pumped back up into the axon terminal, and then it gets reconverted to acetylcholine. And then the acetate is going to be eliminated, right? We're going to get rid of that. Okay, so that's the sequence of events. You should be able to tell that little story. I have everything written down for you. Again, the action potential arrives at the axon terminal. The next thing that happens, the calcium diffuses into the axon terminal. That causes the synaptic vesicles to migrate down. They fuse with the cell membrane of the axon terminal, and the synaptic vesicles now open up and release the neurotransmitter into the synaptic cleft by exocytosis. Then the neurotransmitter will diffuse across the synaptic cleft and bind to the receptor on the postsynaptic membrane. Again, once that receptor receives the acetylcholine, sodium-gated channels open and sodium comes in, causing that local current. And now the action potential, or not, I take it back, the local current travels along the dendrite or the cell body of the second neuron until we get to the axon where the action potential will be initiated. Once the presynaptic neuron stops firing, we want to get rid of any acetylcholine that's in the synaptic cleft, because as long as acetylcholine is in the synaptic cleft, it will continue to stimulate the receptors. Excuse me. So we have an enzyme called acetylcholinesterase. Whenever you see the suffix ase, that means we have an enzyme. Okay, so that's going to, in this case, break down the acetylcholine. And again, like I said, we can recycle the choline part. We use that over again to make new acetylcholine. Okay, so this is a new concept. I've kind of always, this whole semester talked about neurotransmitters causing excitation. That's not always the case, right? We actually have neurotransmitters that can cause another cell to become inhibited. So I just want to talk to you real quick about that. Excitatory neurotransmitters cause depolarization of the postsynaptic membranes, and they promote the formation of the action potential, the nerve impulse. However, inhibitory neurotransmitters, they cause hyperpolarization. They cause the inside of the cell to become more negative, further away from threshold. When some cell is hyperpolarized, it is much more difficult to get it to fire. So in this case, it's going to be inhibited, right? We're going to suppress the action potentials, right? So some neurotransmitters cause excitation, while the others cause inhibition, right? They can actually inhibit a neuron. The effect of the neurotransmitter on the postsynaptic membrane depends on the property of the receptor, not on the neurotransmitter itself, right? You'll see that just, you don't have to write this down, but just as an example, at the neuromuscular junction, acetylcholine is excitatory, but in parts of the brain, it's inhibitory. And it has nothing to do with the acetylcholine, it has to do with the receptor. Okay, so that's the sequence of events at the synapse. The last part of this lesson and the last part of neurophysiology is information processing, right? This is the response of the postsynaptic cell, right? So it's not just being synapsed upon by one other neuron usually. It could have, many neurons synapsing on it. And some neurons may be sending down excitatory signals, other neurons may be sending down inhibitory signals. So now this postsynaptic cell has to process all that information and see if it's going to fire or be inhibited. That's what we're talking about, right? At the simplest levels, right, many dendrites receive neurotransmitters messages simultaneously. Right? It's getting information from a lot of different other neurons. Again, some could be excitatory, some could be inhibitory. The net effect on the axon hillock determines if the action potential is going to be produced or not. All right, so we have to do something called summation, which I'll talk to you about. Postsynaptic potentials, right? Graded potentials developed in a postsynaptic cell are in response to the neurotransmitter. So we said that before. Once the neurotransmitter gets to the the receptor will have the graded potential take place in the cell. So we kind of know that already. So when the cell membrane becomes excited, meaning that it's going to fire, we call this an excitatory postsynaptic potential. We usually abbreviate this EPSP. This is a graded depolarization of the postsynaptic membrane. Right? This is what we've seen so far in all the other lessons I've taught you. This is the new part. There can also be IPSPs. These are inhibitory postsynaptic potentials. Here, graded hyperpolarization takes place. So instead of having us going towards threshold, we're actually going further away from threshold, more towards minus 90, right? Not more towards minus 60 or minus 55. We're going more towards minus 90. We're becoming more negative. So as a result, a neuron that receives many IPSPs is going to be inhibited and it's not going to develop an action potential. And the reason is it's further away from threshold. And it's going to be further away from threshold. To trigger an action potential, right, one EPSP is not enough, right? We're going to need to have more than one signal, hitting our postsynaptic neuron. So in order to do that, we're going to have something called summation. There's two types of summation. We're going to take a look at temporal and spatial summation. I think I do, hang on one second. Yeah, I think I do spatial summation first. So I'm going to go a little bit out of order. Temporal summation is a rapid, repeated stimuli at a single synapse. So all I'm saying here with temporal summation, and I'll show you a picture, if I have one neuron firing repetitively at another neuron, that would be temporal summation. But with spatial summation, I have one postsynaptic neuron that's being fired upon by many different neurons. That is known as spatial summation, right? This is the simultaneous stimuli that arrive at multiple synapses. And I'll show you a picture. So let's take a look at spatial summation first. Spatial summation occurs when two stimuli arrive at the same time, at least two, right? A lot of times it could be more, but at different locations. Local currents spread the depolarizing effects, and the areas overlap experience the combined the effects. Watch this. So would everybody agree? Here's an axon coming to the neuron right here at the, let's say at the, near the axon hillock, and then here's another axon. Both of these are sending signals down. So this is a form of spatial summation, right? This is sending a signal, this is sending a signal, and say both of these are EPSPs, they're causing EPSPs here. That will bring the axon hillock and the initial segment closer to threshold, and now we'll get an action potential that's going to travel down the neuron. Okay, so spatial summation is, again, more than one neuron firing on another neuron. This is from a different book. I like this picture a lot. So watch this. Here is the postsynaptic neuron, and then all these other neurons here are coming in, sending signals down at synapses toward our neuron here. But notice they're not all excitatory. This one down here is sending down excitatory signals, so we develop an EPSP here. We're going closer towards threshold. But this neuron is doing an IPSP, further away from threshold. EPSP, IPSP, EPSP. So what this neuron now has to do is spatially summate, and we add up pretty much all the EPSPs compared to the IPSPs. And if we have more EPSPs, this neuron will fire. If we have more IPSPs, this neuron will be inhibited. Okay, and that's what spatial summation is, right? That is being the postsynaptic neuron is receiving input from many other neurons, and then it has to, again, summate, right? Add all the EPSPs and the IPSPs, and then it will either fire, meaning that it will be excitatory at the trigger area or the initial segment, or it will be inhibited and it won't fire, right? And that's spatial summation. Again, here's just another example. two neurons firing at this third neuron here. And you can see, neuron number one is causing us to get to closer to threshold, and now we reach threshold. This book is saying minus 55 for threshold. So once we reach threshold, because these are firing and we get to threshold, we now get depolarization and repolarization at the initial segment. With temporal summation, we have, where is it? Right here. One neuron firing at the this neuron, and then it fires again, and then it fires again, and then if we get to threshold, it now will cause an action potential, and then the action potential travels. So the concept with temporal summation is we have one neuron repetitively shooting at another neuron. So if you look here, right, this neuron is firing on this neuron. So it fires once, we get, a graded potential that goes up a little bit. It fires again, and then again, and again, and again, and again, and then we get to threshold, and then it fires, right? These would all be like sub-threshold stimuli. I talked about that in a previous lesson. But the concept is that one neuron is firing repetitively at another neuron. That is temporal summation. So let's summate. Let's kind of summarize this. Here's the effects of summation. If the net effect leads to threshold, then an action potential is generated. So all I'm saying, whether it's temporal summation or spatial summation, if we get to threshold, that minus 55, then an action potential will be generated. If the net effect of all the stimuli leads to hyperpolarization, meaning that instead of the curve going up towards threshold, instead we have the curve go down, right? For some reason, the neurotransmitter caused potassium to leave the cell, and the inside of the cell becomes more negative. The curve goes down. The cell will be in hyperpolarization. That neuron will be inhibited, right? It has more IPSPs. Okay, so this is further away from threshold, so this neuron is going to be inhibited. Here's a new term. If the net effect leads to a sub-threshold potential, meaning that the curve is starting to go up, but it hasn't reached threshold yet, we say the neuron is facilitated. It's not quite at threshold, so we say that it's facilitated. If a neuron is in a facilitated state, this neuron will be easier to generate, get it to threshold, right? It'll be easier to excite, meaning get it to threshold where it can then fire. If there are no subsequent more stimuli, then it would just kind of that facilitated state would go away, and it would just go back to being RMP. But when a neuron is, I kind of think of it being like charged, right? It's almost that threshold. It's not quite there. All it would need is another couple stimuli that we say the neuron is then facilitated. Okay. And that is summation, right? So that concludes our lessons on neurophysiology.